Thank you for joining today. I'm Kellie N. Smith, Associate Professor of Oncology and Director of the PhD and TCR Immunogenomics Core at the Bloomberg-Kimmel Institute for Cancer Immunotherapy. I wanted to give a huge thank you to the folks at LabRoots for inviting me to speak here today, and I'm very excited to share our work with you. Here are my disclosures. I always like to start my presentations with a little dose of reality in terms of the public health concern of lung cancer. Around 1 in 16 Americans will be diagnosed with lung cancer in their lifetime. 10 to 15% of these are in patients who have never smoked, and it can affect anyone regardless of gender or ethnicity. It affects men and women around equally. 361 Americans die of lung cancer every single day, which is the equivalent of a commercial airline jet crashing every single day. And if this was happening, it would be a huge public health concern in our country. And that's why lung cancer is really a major issue that we need to address. Not only is lung cancer a major public health concern, but it's also a significantly underfunded public health concern. Lung cancer takes more lives every single year than the next most prevalent 3 cancers combined. So lung kills more people than prostate, colorectal, and breast cancers combined. Yet it only receives 6% of federal research dollars. It kills 1.4 times as many women as breast cancer does, but it receives significantly less funding. So it's a huge public health concern, but it's a significantly underfunded concern as well. Recently, immunotherapy has moved into the early stage setting for patients with lung cancer, namely in the format of neoadjuvant immunotherapy. What this means is that patients are eligible to receive immunotherapy for several weeks prior to going to surgery to remove the tumor. And the goal of this is twofold. First is to reduce the size of the tumor prior to surgery to make the chances of success greater. And second is to promote systemic immunity in these patients to either prevent or delay relapse of the disease. And this has really been borne out by several clinical trials that have been published over the last few years. We see that there's improved overall survival in patients who have received neoadjuvant immunotherapy combined with chemotherapy relative to chemotherapy alone. And this has really been a significant benefit in the clinic for patients. But there's also significant scientific value to the neoadjuvant setting, namely that we're able to obtain before, during, and after immunotherapy biospecimens, which is not something that's routinely possible in patients who are being treated for metastatic disease. So patients receiving immunotherapy for advanced lung cancer don't routinely undergo biopsies. So it's really hard to get tissue that has been treated with immunotherapy. But in the neoadjuvant setting, this is extremely doable and extremely possible, and it allows us to really study a wealth of information that we can glean from these biospecimens that are obtained before, during, and after treatment. Even though immunotherapy has been immensely successful in the early stage setting, the fact is that most lung tumors still do not respond to immunotherapy. And we know that PD-L1 and tumor mutational burden are imperfect biomarkers. So in order to better develop biomarkers and therapies to overcome immunotherapy resistance, we really need to understand the endogenous immune response. So we need to better understand the cells that are being directly targeted by immunotherapy. So there are very few fundamental truths in immunology, but one of them is that in order for PD-1 blockade to be effective, the patient must have a pre-existing anti-tumor immune response. If there is no immune response existing to unleash, you can throw all the anti-PD-1 you want at a patient, but it's not going to do anything. It's not going to work. So we really need to understand these endogenous tumor-reactive T cells if we want to develop these better biomarkers. And some of the few lingering questions are: how can we detect and study these cells? What are the transcriptional programs and targets that are associated with response versus resistance? What other cell types do these tumor-reactive T cells interact with? And what is the mechanism by which these interactions can impact anti-tumor immunity? To address these questions, we started by performing single-cell TCR-seq RNA-seq on tumor-infiltrating lymphocytes from lung cancers that had been treated with neoadjuvant immunotherapy. We identified 15 distinct T cell clusters, and we had data from around 560,000 T cells after stringent quality control. Not surprisingly, when we looked at pseudobulk gene expression between tumor and adjacent normal lung, we were able to separate these specimens using principal component analysis. But when we were looking at response status, basically separating by MPR, which is major pathologic responder, versus non-major pathologic responder, we weren't able to see this distinction. So at the pseudobulk gene expression level, we're actually seeing no differences in our T cells according to response status. So this really provides the rationale for specifically looking at tumor-reactive T cells in these cancers, which are going to be much less frequent. And this fact was evidenced by a paper published by Yannick Simoni and Evan Newell a few years ago, where they profiled 40 cancers for tetramer recognition. And these were tetramers that represented neoantigens or tumor-associated antigens. And from among 1,200 tetramers that they queried, they were only able to identify 2 neoantigen-specific responses, one in each of 2 different tumors. So this led the investigators to conclude, rightly, that the abundance of tumor-reactive T cells in the tumor microenvironment is actually very low, and that the majority of T cells are bystanders that are irrelevant to anti-tumor immunity. And this was very similar to what we were finding in our own data, that these cells are just very hard to detect. So how can we make this process more streamlined? We can't be making 1,200 tetramers for every single study, so can we develop methods to identify tumor-reactive T cells more quickly and easily so that we can facilitate therapeutic discovery? Following our initial publication of our single-cell data from neoadjuvant immunotherapy-treated lung cancers, we discovered that in an analogous data set published by Giacomo Oliveira and Kathy Wu on melanomas, we identified that tumor-reactive cells really tended to localize to similar areas of the UMAP. Now, what we're showing here on the left are tumor-reactive TIL, which are shown in red. So these are specific for MANAs, or mutation-associated neoantigens. These localize to a cluster that we call tissue-resident memory. Whereas in the Oliveira Nature study, the tumor-reactive T cells localize to a cluster they called terminally exhausted. But when we actually look at the genes expressed by these cells, it turned out that they're actually very similar in their gene expression programs. So we call them different things, but effectively these cells looked very similar at the surface. So we had this question of, are there common transcriptional signatures among tumor-reactive TIL from different cancer types? And can we exploit this to better detect these cells across tumor types and in our single-cell data? We therefore ran both of these data sets through a standard pre-processing and quality control pipeline and integrated them into the same UMAP. And what we can see, as shown here, is we're really seeing localization of the tumor-reactive cells, or neoantigen-reactive cells, which are shown in red, in similar locations in the lung cancer data, which is on the left, as in the melanoma data, which is in the right. So this shows us that even though in the original publications of these data sets, these cells were labeled as 2 distinct kind of cell compartments, when we actually integrate the data and overlay them, they tend to localize to pretty similar regions of the UMAP. And when we dug a little bit deeper into which genes are being expressed by these cells and which genes are prominent in this region of the UMAP, we found that these cells express high levels of CXCL13 and CD39. So CD39 was identified in the Newell paper with the tetramers. But we also identified high expression of CXCL13, and these cells also express very low levels of the IL-7 receptor. So this prompted us to develop a 3-gene signature that could potentially identify tumor-reactive cells based on single-cell transcriptomics data alone. Zhen Zhang, a junior faculty member in the lab, really led this effort, and she started by using a discovery and test cohort to optimize the model that we now call MANASCORE. We then tested this model on several publicly available data sets as a method of independent validation. And as you can see here, we have various combinations of test and validation data that we were able to use. Now, it should be mentioned that the amount of transcriptomes that have been published on tumor-reactive TIL is relatively low in terms of machine learning standards. But we used what was available to us. And what you can see is that when using imputed and non-imputed data, the MANASCORE, which is shown in the red and the blue lines here, either outperforms or performs equally as well as the neo-TCR8 signature that has been published previously. And we see this in our test data as well as in our validation data set. So this really was exciting to us to indicate that a 3-gene signature could perform as well or even better than the neo-TCR8 signature, which has over 200 genes used in it. So the benefit of using the more parsimonious 3-gene signature is that it then opens up the possibility for multispectral IF studies, as well as moving it into the clinic as a potential biomarker. One thing that makes our score unique also is the use of patient-specific thresholds to identify MANASCORE high cells. As you can see here, we really have a range of MANASCOREs within individual patients. So just to orient you, the x-axis is each individual clone that is found within the tumor of these two patients. So we have MD01005, MD01010. These are two different patients. And then on the y-axis, we have our overall MANASCORE, which is calculated using the imputed and non-imputed values from the gene expression data. And you can see that within each clone, there are many cells, and there's a wide range of MANASCOREs that each of these cells have. And so not only is there intraclonal heterogeneity, but there is heterogeneity within individual patients, right? Some of these clones have higher MANASCORes than others, but then you also have interpatient heterogeneity, so between different patients. And I should mention that MD01005 was a patient with 0% residual tumor, so this was a complete responder. So it's not like the MANASCORE is some indication of the clinical response, because we would expect this patient to have a higher MANASCORE. It really just boils down to individual patient biology. So by leveraging this, we're able to set patient-specific thresholds, and I have one example here of a patient where we plot the non-imputed MANASCORE versus the imputed MANASCORE, and we set a threshold for each patient based on the last trough in the MANASCORE distribution. So basically what we're showing here is all of the MANASCORE high cells. To identify putative tumor-reactive cells, we then take the T cells that share a TCR with MANASCORE high cells. So within a TCR clone, as you can see here, you can have some cells are MANASCORE high, some cells are MANASCORE low, but they all recognize the same antigen because they share the same TCR. So if we identify cells that pass a threshold of number of cells that are MANASCORE high, we will identify all cells with the TCR as a putative tumor-reactive cell. We then wanted to compare the localization of cells that have been validated functionally as being tumor-reactive and see if they co-localize with our identified putative tumor-reactive cells, or PTRCs, as identified by MANASCORE. So here I'm showing the functionally validated neoantigen-specific T cells from our integrated lung cancer and melanoma data. And when we overlay our putative tumor-reactive cells, we can see almost near identical overlay of PTRCs identified by MANASCORE with those cells that have been functionally validated as being tumor reactive. We next wanted to validate this on other publicly available data sets. So we accessed the oral cancer data set from Luoma et al. And this data set was interesting in that it also contained endogenous retrovirus specific T cells and T cells specific for HPV antigens. So we mapped the validated tumor reactive T cells from this study onto their UMAP. There were 136 cells. And when we overlaid Manascore, we can see direct colocalization of Manascore high cells with the validated tumor reactive cells. This was also a similar trend when looking at the Lowry metastatic cancers data set. So this was from a variety of different cancer types, where we have the validated cells on the left and then near identical overlay of our Manascore on the right. We then wanted to leverage a novel data set of Merkel cell carcinomas. So the majority of Merkel cell carcinomas are associated with the Merkel cell polyomavirus. Their mutational burden is very, very low, and it's thought that the vast majority of the immune response is directed to the large T antigen from the Merkel cell polyomavirus. So we generated a data set using site-seq tetramers coupled with single-cell TCR-seq RNA-seq to identify tumor-reactive T cells. So these are T cells that are specific for Merkel cell polyomavirus using multimers that were specific for known immunodominant epitopes in this virus. We identified a large number of tumor-reactive cells using this tetramer approach. Then we overlaid Manascore, and once again, we identified near direct overlay of our Manascore with our tumor-reactive TIL. And in fact, there were only 42 cells, or 6 TCR clones, that were not captured by our Manascore. And this was really striking and interesting to us because not only is Manascore able to identify tumor-reactive cells across different tumor types, but it also seems like Manascore is able to identify T cells that are specific for different classes of tumor antigen. So we're able to identify T cells that recognize TAAs, neoantigens, endogenous retroviruses, as well as viral oncogenes, which is something that has really not been assessed previously. We then wanted to use Manascore to validate and discover new biology that's relevant to immunotherapy response. So we first just looked at the proportion of putative tumor reactive cells in the tumors, and we saw that there is a significant difference in the percentage of PTRCs in responders versus non-responders. And this is correlated, the proportion of PTRCs in the tumor is negatively correlated with percent residual viable tumor at the time of surgery. So we are seeing a very nice association between the tumor-reactive T cells and the clinical response in these patients. We then performed differential gene expression analysis of PTRCs in responders, which is shown on the left, versus non-responders, which is shown on the right. In our responder PTRC, we saw high expression of granzyme K. We also saw higher expression of CXCL13 and IL-7 receptor, even though these are two molecules that are in the Manascore itself. So even though these are IL-7 receptor low by nature, they express higher levels than in our non-responding tumors, which had high expression of things that we canonically associate with cytotoxic effector function. So things like granzyme B, granzyme A, granulysin, interferon gamma, and some killer-like receptors. And although this was quite shocking to us at first, what we've since found is that these effector molecules a lot of times can hang around transcriptionally for long after these T cells have become exhausted of their effector function. So more often in the context of cancer, co-expression of a lot of these effector molecules with also checkpoints such as LAG3 and TIGIT really confers more of a terminally exhausted or dysfunctional phenotype. Whereas in our responder PTRC, we're seeing things like EOMES, CCR7, IL-7 receptor, and granzyme K, which are all things that are associated with a non-exhausted or even a potentially progenitor-exhausted phenotype of cells that are still highly proliferative and can respond to immunotherapy. We next wanted to understand if our putative tumor-reactive cells interact with other cell types in the TME. And the easiest thing for us to start by looking at was regulatory T cells, since these are known to interact with CD8 T cells in tumor microenvironments. These are known to be a suppressive mechanism in the context of immunotherapy, and we already had the transcriptomic data from when we generated our large single-cell data set of 560,000 T cells. So the first thing we did was we identified these subsets in regulatory T cells that were had more evidence of being immunosuppressive, and we identified this activated 1 subset, and this was published a few years ago in Science Immunology. And this activated subset had high expression of TNF receptor superfamily members, so we have GITR here as well as OX40. And these molecules were highly upregulated, and these are known to mark highly activated Treg subsets. And when we looked as a function of response, what we saw is that this activated 1 subset was much higher in non-responding tumors relative to responding tumors. One thing that was also particularly interesting, if we think back to our tumor-reactive T cells, we recalled that OX40 ligand was expressed by tumor-reactive CD8s. So when we queried the ligand for OX40, so OX40 ligand, on our tumor-reactive TIL, we saw that non-responding T cells had much higher expression of the ligand for OX40. So this indicated to us that there could potentially be direct interaction of our tumor-reactive cells with these highly immunosuppressive regulatory T cells in the tumor microenvironment. One other thing that we noticed about this activated 1 subset is that they also expressed high levels of CCR8. CCR8 is another molecule that is being targeted clinically and is known to mark a highly suppressive subset of regulatory T cells. So we have our OX40, which is marking activated Treg, and then we have CCR8, which also marks suppressive Treg. And we had this question of how OX40 and CCR8 kind of interact, or if they even do interact, as a function of immunotherapy response, and how these cells interact with tumor-reactive CD8s directly. Now, even though regulatory T cells represent an enhanced abundance in tumors relative to adjacent normal tissue and relative to the periphery, there are still relatively few Tregs that we're able to get from digesting tumors. They make up a small proportion of the cells in the tumor, but they are extremely potent. But in order to profile these with a lot of rigor, we wanted to generate a Treg single-cell data set with many more cells than what were available from our lung cancer data alone. So we had around 75,000 tumor-infiltrating Tregs from our lung cancer data. And we wanted to then merge this with several other data sets that we had at our disposal. So 2 of these were from neoadjuvant immunotherapy studies in head and neck cancers that we had generated in our lab. And then there was one that was published in Cancer Cell this year that was also in head and neck cancers treated with neoadjuvant immunotherapy. So we had 4 data sets of neoadjuvant immunotherapy in lung and head and neck cancers that had single-cell transcriptomics from regulatory T cells. So we obtained the raw data from all 4 of these data sets and processed them with our standard pre-processing pipeline and integrated them into a single UMAP. And as you can see here, we were able to get around 141,000 tumor-infiltrating regulatory T cells, which is a very large number of Tregs that we're able to profile at the single-cell level. And when we look at some of the key transcriptional signatures that we identified previously in our lung cancer data, we can see that high expression of these TNF receptor superfamily members, OX40 and GITR, as well as CCR8, is really restricted to this activated cluster up here, which is analogous to our activated cluster we identified previously that is associated with immunotherapy resistance. But what we can see is that CCR8 is really just expressed at the top portion of this cluster. It's not really globally expressed in the same way that OX40 is. So we wanted to understand what is the expression pattern of CCR8 as it relates to OX40. So we separated our tumor-infiltrating regulatory T cells based on CCR8 and OX40 expression. And what we can see is that the vast majority of OX40 positive regulatory T cells actually don't express CCR8. It's really just a small subset of Treg that are double positive for both of them, whereas nearly half of our Treg express OX40 without CCR8. So this really opened some additional questions for us related to the suppressive nature and the transcriptional programming of these cells. Namely, we wanted to know if the suppressive function of the OX40 positive subset that we identified previously is actually linked to OX40, or if that's more a function of the CCR8. So we next performed differential gene expression between our CCR8 negative OX40 positive Tregs, which is shown on the left, and our CCR8 pos OX40 pos Treg on the right. So we have Treg that only express OX40 here, and Treg that express both of these molecules over here. And what was very striking to us is that when you separate these cells in this manner, you see a lot of genes that have been previously associated with regulatory T cell function. So we have CTLA4, CD39, Lalin, TIM3, and 41BB, which we kind of refer to as the usual suspects of Treg markers. These are very well-defined markers of activated and suppressive regulatory T cells. But then we also identified some lesser-known players. So in the Treg biology world, these aren't lesser known, but these are perhaps not as highly understood as some of the other markers. So EBI3, which is part of the IL-35 heterodimer, which is a newly discovered cytokine within the last, I would say, 10 years or so, that is associated with suppressive Treg function. CD177 and CCL22 have both been identified as being upregulated in suppressive CCR8 positive tumor infiltrating Treg in other studies. And then IL-1 receptor 2, which is actually a decoy receptor for IL-1. So this receptor binds to IL-1 in the tumor microenvironment and sequesters IL-1. So we don't actually get IL-1 signaling, but it sequesters IL-1 away from other T cells in the tumor microenvironment. Where, interestingly enough, if we actually look over at our Treg that express OX40 but do not express CCR8, we see upregulation of sphingosine phosphatase receptor, CCR7, TCF7, so things that tend to be associated with either lymphoid homing or resting regulatory T cells. We don't usually associate these molecules with a highly suppressive subset. So this really indicated to us that it really was the CCR8 expression that was driving this suppressive phenotype and perhaps the association with ICB resistance rather than the OX40 expression. And indeed, when we take a look at the suppressive activity of these cells ex vivo, we see a similar finding as we do with our transcriptional programming. So we sorted out Treg from untreated tumors that were either double positive for OX40 and CCR8 or were OX40 pos CCR8 negative. And what we can see is that even at high Treg to T conventional T cell ratios, it really is just the double positive Treg that are able to suppress proliferation of conventional CD4 positive T cells. So this really supports the notion that it is the CCR8 positive cells when expressed in conjunction with OX40 that are facilitating this suppression. Unfortunately, Treg that express CCR8 but not OX40 are so infrequent in the tumor, they make up less than 5% of Treg, that it really was impossible for us to assess this subset ex vivo. We next wanted to assess the potential for direct interaction of our Treg subsets of interest with our putative tumor reactive cells in the tumor microenvironment. And one issue that we run into a lot when trying to compare spatial interactions between different tumors is how do we normalize for these distances? But one thing that's really cool in the neoadjuvant space is that you often have these resected tumors that have mixed treatment effect within the same tumor specimen. So you can actually compare a region of response versus a region of resistance within the same tumor. So that allows your analysis and your comparison to be much more robust. So that's exactly what we did here. We chose a ICB-treated resected lung tumor that had one area of immune-mediated regression, as well as an area with a residual tumor nest that had no evidence of treatment effect. And we compared the nearest distance of our OX40-pos CCR8-positive Treg to the nearest Manascore high spot, or the nearest putative tumor reactive cell. And what was really cool to us is that if we look in the tumor nest, so this is the area that was ICB resistant, so there was no evidence of ICB mediated tumor regression, these double positive Treg are much closer to our putative tumor reactive cells. Whereas if we look in our area of regression, these cells are much further apart, suggesting that this response at the pathological level could be associated with the interaction of our double positive Treg with our tumor reactive T cells. And since immunotherapy directly targets our tumor reactive T cells by blocking PD-1 on their surface, it is conceivable to think that these OX40 positive, CCR8 positive Treg could also be negatively impacting immunotherapy response as well. So we took a look at our merged data sets of four types, or four neoadjuvant studies of immunotherapy in lung and head and neck cancers. And what we saw is that in non-responders only do we actually see an increase in the proportion of these double positive Tregs during immunotherapy treatment. So you can see this stark increase in the percentage of these Tregs in the tumor from before versus after immunotherapy. Whereas in responding tumors, we actually see the opposite trend. We actually see these regulatory T cells going down in frequency at the post-immunotherapy time point. And what's really interesting too is that if you just compare the blue bar versus the blue bar, I'm not showing the statistics here, but prior to treatment, there actually is no difference in these subsets. So it's not just a matter of having an endogenous difference in your double positive Tregs prior to immunotherapy. It actually seems like immunotherapy is inducing these changes in some patients rather than others. And we also see a similar trend when we look at the correlation of this double positive population with the percent residual viable tumor at the time of surgery. So the more double positive regulatory T cells that you have, the more residual tumor you have after immunotherapy treatment. So this really indicates to us that not only are they potentially negatively influencing the tumor reactive T cells, but it also seems like immunotherapy in some patients is actually initiating this expansion of regulatory T cells, and that this could be a mechanism of resistance to immunotherapy in the resectable setting. The obvious question then becomes, what is causing this difference in expansion kinetics as a result of immunotherapy treatment? The first obvious thing that we thought could be playing a role is either PD-1 or PD-L1 expression on regulatory T cells or on antigen-presenting cells, such that when you block the PD-1, PD-L1 interaction, it causes some different levels of expansion. So when we queried our regulatory T cells for PD-1 expression and we queried non-T cells for PD-L1, we didn't see any difference prior to immunotherapy. And I'm not showing that data here, but that indicated to us that at least the level of expression of these molecules was not responsible for the differences that we were seeing in the double positive expansion in non-responders versus the retraction in the responding tumors. So the next thing we thought about was to look at ligand expression for CCR8. Now, the dominant ligand for CCR8 that we all think of in the field is CCL1. So we took a look at CCL1 in our spatial transcriptomics data, and CCR8 as well. And what you can see and what you can appreciate here is we see very nice CCR8 expression in our tumor nest, as well as in our area of regression. But when we took a look at CCL1, we actually could find basically no expression of CCL1 in our spatial transcriptomics data. Now, this could be a function of the technology. So we queried CCL1 in our single cell data as well, and again found basically no expression of CCL1 in either T cells or non-T cells. And again, this could just be a function of transcriptomics. So we do have studies underway looking to test for CCL1 at the protein level. But something else that was a little bit encouraging is that CCL18, which is another CCR8 ligand, is actually highly expressed, and we are able to detect this at high levels in both our single cell and our spatial transcriptomics data. Now, CCL18 is a lesser studied ligand for CCR8, possibly because the mouse ortholog, which is actually CCL8, there is no CCL18 in mice. So that's potentially the reason why it has been a little less studied. But in lung, at the very least, CCL18 is very highly and constitutively expressed. And we see this very nicely in both our tumor nests and in our area of regression. We see high levels of CCL18. So this was very striking to us, and now this is really created a new area of investigation for our lab, is looking at the role of CCL1 versus CCL18 in attracting these CCR8 positive Tregs into the tumor microenvironment. In conclusion, what I have presented to you all today is really our integrated analysis of tumor-reactive CD8s and regulatory T cells in the context of resectable cancers treated with neoadjuvant immunotherapy. I've discussed MANA-score, which is a 3-gene signature that can accurately identify tumor-reactive CD8-positive TIL, and we've shown that this score can be used to discover new biology and associations with clinical outcome. More than half of the OX40 positive regulatory T cells that we find in our data are CCR8 negative, so the vast majority of OX40 positive Treg actually do not express CCR8, and this is consistent across cohorts and across different tumor types. But what's really interesting is that CCR8 is largely restricted to the OX40 positive regulatory T cells. So remember, less than 5% of Treg express CCR8, but not OX40. So this is something we really want to explore in the future. We found that CCR8 positive Treg are uniquely immunosuppressive, and that OX40 by itself does not confer this suppressive function. And we want to better understand this mechanism. Potentially it's through IL-1 sequestration, as evidenced by upregulation of the IL-1 receptor 2, or some other mechanism, either TCR dependent or independent. We also found that the double positive Tregs increase in our non-responding tumors during immunotherapy, but we actually see them decreasing in our responder tumors. And this was a very interesting observation for us. And we found the evidence that these double positive Treg could interact with our tumor-reactive CD8s, as evidenced by the OX40 ligand expression on our CD8s. So moving forward, we are currently focused on optimizing MANA-score for CD4 positive TIL, since we know that CD4 positive T cells really play an integral role in anti-tumor immunity and in the response to immunotherapy. We want to further understand why these Treg, especially the double positive populations, are responding differently to immunotherapy. So we see them increase in non-responders and decrease in responders. We really want to understand what is behind that. What is the mechanism that's promoting that? We think it could be through some modulation of CCR8 ligand expression. So is PD-1 blockade somehow enhancing or not the expression of CCR8 ligands? And then we also want to know what is the relationship between Treg that express varying levels of OX40 and CCR8. Are they all part of one linear differentiation trajectory, or are they representative of distinct differentiation pathways? And this is something that we're currently exploring. I would like to thank all of the members of our lab and all of our collaborators who made this work possible. Specifically, most of the data I presented here today was generated by Arbor Dikema, Sung-Soo Man, Zhen Zhang, or Tia Zhang. And all the members of my lab contributed to this work in one way or another, so I want to thank all of them. From medical oncology, I really want to thank Patrick Ford and Julie Bramer for working tirelessly to get us these biospecimens for patients enrolled to these trials. Our collaborators in cancer genetics and biology, especially Elsa and Ignastu, and the entire biorepository team who helps us acquire these specimens. Hongkai Ji is really our biostatistics and bioinformatics guru, who leads development of a lot of these computational pipelines that we use here. Our pathology collaborators, Janice Taub and Julie Stein-Deutsch, as well as our funding, and of course the patients and their families for making all of this possible. And once again, I do just want to extend a huge thank you to the folks at LabRoots for inviting me to give this presentation today. Thank you.